So far, we have learnt why surface science is important, and how to make a new surface on an object, either by deposition methods such as spin coating or painting, or by chemically bonding new species on, such as in plasma treatment. Now we will look at how these methods are being used to functionalize surfaces to make them biocompatible. But what is biocompatibility? It can be described as the ability of an object to coexist with living tissue or biological organisms without causing harm. The surface chemistry of biomaterials play a hugely important role in determining whether cells in the body will successfully respond to an implanted medical device and promote wound healing, or whether the body will have an unhealthy reaction to it. These reactions could be inflammation, infection, fibrosis, or thrombosis. The use of biomedical implants or foreign bodies span right across the field of medicine, amplified also as a result of the aging population. From contact lenses to dental implants to hip replacements, the research going on in the field of surface chemistry to improve the safety and functions of these devices would have a huge impact right across the world. To understand how to make a surface biocompatible, we need to know how the body responds to implants in the first place. When any foreign object is put in contact with human tissue, the first response between blood and biomaterial involves adsorption of proteins onto its surface, hence developing a blood-based matrix around the material which the cells respond to. The success of the implanted material within the body is largely down to these adsorbed proteins. Even when the implant is non-toxic, unreactive, and non-immunogenic, inflammation and rejection can still occur. This is often because it is actually the conformation, or shape, that the proteins take on when they adsorb to the surface, which is triggering the bad immune response. Sometimes, when the proteins adsorb, they change conformation and shape, exposing hidden parts of the proteins whose role is to specifically trigger inflammation. So how can we modify the surface of a material to alter protein adsorption? For biocompatible surfaces, the objective is to either prevent protein adsorption onto the surface entirely, or to control which proteins adsorb and their adsorption conformation. Two ways to achieve this is adding functional groups to the surface to make it have certain properties, or to attach proteins or biological molecules onto the object prior to implantation for control over what adsorption may take place. We will start with adjusting the surface properties, such as by making a surface more hydrophilic, which means having a high affinity for water molecules. Although all proteins have different kinetics, in general it is thought that if a surface is made to be hydrophilic, then water molecules want to be near the surface more than the proteins do, so water outcompetes with the proteins and blocks them. more protein-resisting surface properties have been found. As well as hydrophilicity, protein-resisting surfaces are those which contain electronically neutral groups, and those which take part in hydrogen bonds by being the acceptor but not the donor. For example, electronegative oxygen and nitrogen atoms with no electropositive hydrogens. Another way to improve biocompatibility is to attach proteins or biological molecules onto the surface before implantation, so that when the implant is in the body, there are already proteins in the desired conformation to interact successfully with cells and blocking further unwanted protein adsorption. Grafting can be used, which involves activating the surface with methods such as UV radiation, plasma, or chemical reactions, so that proteins can thus react on. The limitations with this technique are that the proteins immobilized will be less mobile, and it can be difficult getting them in the correct conformation. Self-assembled monolayers are formed by spontaneous chemisorption of molecules onto a surface, and as the terminal group at the end of the organic chain can be adjusted, a functional group can be chosen onto which proteins would bind. Self-assembled monolayers provide an additional high degree of control into density and the conformation taken on by the chemisorbed molecule, so hence with the proteins also. However, the surface which the molecules chemisorb onto must be gold or silver coated, so it is unsure how popular this technique will be due to this limitation. Plasma polymerization can be used to produce polymer films onto an object, which either have functional groups known as coupling groups, which proteins can react with and hence be immobilized, or films which have properties that are able to resist non-specific protein adsorption, such as hydrophilicity, as mentioned earlier. Plasma polymerization is arguably the most promising technique due to its compatibility with most materials, and its uniform and conformal treatment to all surfaces on the whole object. The research going into the field of surface chemistry for improved biocompatibility of objects is very widespread, and has even developed a new field called tissue engineering. Scientists are researching how to grow new body tissue in the lab using stem cells. Stem cells are unspecialized cells and can convert into many different types of cell to grow new tissue, such that they can be implanted and be used to repair the damaged system. This technique can aim to produce life-saving replacement organs, or heal tissue which is damaged and hence drastically decreasing people's quality of life, such as tissue on their spinal cord. Biocompatibility in this field is extremely important, because they need to use a polymer scaffold to grow the cells on to create the new tissue in vitro. However, whilst the methods described earlier involving resisting non-specific protein adsorption are useful for foreign objects which will be providing support within the body, such as dental implants, In the field of tissue engineering, where cells are required to interact fully with the scaffold and let it integrate into the cell's matrix, this opens up a multitude of new challenges for the future. In summary, we have briefly looked at why surfaces need to be biocompatible, and two methods being used to make them so, which are altering the properties of surfaces by having certain functional groups present to resist non-specific protein adsorption, or by immobilizing proteins or other biomolecules onto the surface in the correct conformation. We have looked at situations where biocompatibility is really important, making it clear that surface chemistry research is providing a platform for solving so many medical problems worldwide.